Good morning. It's March 25th, and this is your daily brief in science. Here's everything you need to know. A 600-year-old grape seed found in a 15th-century Valenciennes hospital toilet links today's Pinot Noir directly to medieval practice, a finding that reshapes our view of wine history. DNA analysis shows extensive ancient trade and mixing of domesticated grapes with local wild vines, with exchanges spanning Spain, the Balkans, the Caucasus, and the Middle East during Roman times. France's central role in wine history emerges, highlighting how cloning and propagation spread major grape varieties around the world. A long-running Japanese study shows a hard limit to mammalian cloning. Over 2 decades, researchers cloned more than 1,200 mice across 58 generations using nuclear transfer, and the 58th generation died shortly after birth, marking a practical ceiling for this method. Early generations improved, but by generation 25, chromosomal abnormalities appeared and the X chromosome was lost, with mutation frequencies nearly doubling by generation 57. The team conducted more than 30,000 cloning attempts to test whether a single lineage could be indefinitely cloned through asexual reproduction. The authors say accumulated mutations could generalize to vertebrate cloning and have major implications for agriculture and animal breeding that rely on cloning to preserve genomes. The results align with Muller's ratchet, where deleterious mutations accumulate in asexual lineages, risking mutational meltdown and possible extinction. While methods to improve cloning continue, there is no clear solution to prevent meltdown, and some teams are pursuing non-lethal cell collection techniques to aid endangered species recovery. The study was published in Nature Communications and led by geneticist Sayaka Wakayama. Findings suggest a practical limit to cloning across generations and imply that sexual reproduction may reset mutation load, offering insight into why sex persists in mammals. Even when clones appeared normal at birth and had ordinary lifespans, genetic changes accumulated each cycle, increasing the likelihood of future cloning failures and limiting long-term viability. The overarching takeaway is that repeated mammalian cloning drives irreversible genetic decay, underscoring the essential role of sexual reproduction in maintaining genome integrity. Clones were produced using nuclear transfer, the same method as Dolly and Cumulina, with cumulus cells serving as the source, reinforcing the technique's limits observed in this long-running study. A new United Nations-backed assessment warns of a freshwater fish crisis, with 325 migratory species needing international protection coordination to prevent extinction. The scope is broad. 325 migratory freshwater species are prioritized for coordinated action under the CMS, and 97% of migratory species are already at risk, with Asia alone reporting a more than 95% decline in megafish since 1970. Overall, migratory freshwater fish populations have fallen about 81% over the last 5 decades, driven by dams, pollution, and overfishing. Conservation strategies include keeping rivers free-flowing, reconnecting fragmented waterways, improving fisheries management, protecting floodplains and wetlands, and restoring degraded habitats, with examples like removing dams on the Elwha and White Salmon rivers reopening habitat for multiple species. The issue is being discussed at the CMS COP in Campo Grande, Brazil, where governments are weighing expanded protections, reduced habitat loss, and stronger anti-poaching and habitat conservation measures for migratory species. Countries along migratory routes are considering cross-border conservation plans to establish defined migratory corridors and joint quotas for species such as an 11,000-kilometer whales and other migratory fish. In the Mekong Basin, fisheries yield exceeds 2 million metric tons annually, sustaining tens of millions, so losing migrations would threaten food security and cultural practices. Decline is driven by dams, habitat fragmentation, pollution, overfishing, sand mining, and climate-driven changes that disrupt long-connected river corridors essential for migrations. Barriers like dams and culverts fragment rivers, alter flows, and hinder spawning grounds, increasing energy costs, disease risk, and predation. Since most migratory fish cross borders, cross-border cooperation is essential to recover national stocks and protect habitats through coordinated government action. Experts urge moving beyond iconic megafauna to emphasize migratory freshwater fish's cultural significance, ecological roles, and economic value. Notable regional examples include the Mekong giant catfish's decline due to dam barriers and overfishing, while species like Tre Riel, Hilsa, Amazon dorado, and other large catfish migrate across continents. Basins affected include the Amazon, La Plata Parana, Danube, Mekong, Nile, and Ganges Brahmaputra. A broader shield forms when people have immunity from prior infection or vaccination against SARS-CoV-2, and this shield could slow or prevent the emergence of a new human-infecting sarbecovirus. A study from the MRC-University of Glasgow Centre for Virus Research analyzes how population immunity to SARS-type viruses might reduce the risk of a future coronavirus pandemic. Researchers say the protective effect can act as a biological barrier, but it is not a guarantee against all threats. The work emphasizes that vaccines are most effective when deployed promptly after the first SARS-CoV-X case is detected, with delays likely reducing impact. Pablo Murcia and Brian Willett describe population-level immunity as a collective defense that amplifies when vaccination campaigns are timely and widespread. The study notes that delaying vaccination could lessen its effectiveness in preventing transmission. Published in Nature Communications, the research examines post-pandemic changes in population immunity and how they could influence the likelihood of zoonotic coronavirus emergence. Funding for the study came from UK research and innovation bodies, the MRC and BBSRC. The findings underscore that vaccination timing matters for maximizing the protective effect against any emerging coronavirus threat. The researchers caution that while population immunity provides protection, it does not eliminate the risk of future coronavirus threats. A drought intensifies the risk of antibiotic resistance in soil and across health systems, and new findings link aridity to higher rates of resistant pathogens. Researchers report that drought and aridity concentrate antibiotics in soil, elevating resistant strains through controlled experiments and metagenomic analyses, with global hospital data showing drier regions harbor more resistant pathogens even after accounting for socioeconomic factors. The study combines global metagenomics, lab-based systems, and epidemiology across more than 100 countries, aiming for consistency in the story that drought may amplify resistance worldwide. Led by Diane Newman and Xiaoyu Shan of Caltech, and published in Nature Microbiology on March 23, 2026, the work situates its findings within a broader antimicrobial resistance literature, citing high-profile sources and supporting reviews. The article notes implications for hospital antibiotic choices may depend on local aridity, underscoring the need for rapid diagnostics, multi-drug therapies, and renewed investment in basic antibiotic discovery. It also highlights policy implications, such as environmental resistance surveillance, cross-sector antibiotic stewardship, and climate-aware strategies beyond clinical settings. The piece emphasizes a One Health approach, integrating environmental monitoring with prudent antibiotic use and climate adaptation. While the Nature Microbiology article is a preview requiring institutional access, the authors and affiliations include Timothy M. Ghali of Macquarie University and the ARC Center of Excellence in Synthetic Biology. The finding aligns with a growing body of literature on antimicrobial resistance and is contextualized by related studies and reviews in Lancet Microbe and Nature Reviews Microbiology. Overall, the warnings point to drought-prone regions like Spain and stress the importance of science communication and sustained scientific journalism in guiding policy and practice. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.